who is bearing the load in these muscles. And so we would do a mechanical experiment where we would take a muscle fiber or take a fiber bundle, which is obviously the muscle fiber plus its extracellular matrix. And then this is maybe 10 or 15 fibers. And then we called it a fiber group, 10 or 15 separated muscle fibers. So very little ECM, and measure the mechanical properties. Because as you all know, one of the tricks in measuring mechanical properties is coming up with material properties and therefore cross-sectional areas. So it can be challenging. But the important finding of this study was that the load bearing, if you actually measure the modulus of the specimen, fibers and groups were about the same. In other words, a group of fibers, a bunch of fibers, was just, a scaled up version of fibers. But the bundles with the ECM were much more stiff. And in fact, it looked like in this system, this was mouse muscle, in this system, only about 10% of the bundle load was borne by the fiber. And, as we were talking in our chit chat, I was raised on frogs. And when you measure frogs, if you look at the relationship between sarcomere length and modulus, we can have that discussion what the right units are. Bundles and fibers are about the same. In fact, as most of you know, this is how Alan Magid figured out that there was titin inside muscle fibers, because the fiber, the bundle, and the whole muscle had about the same modulus. Whereas if you do the same thing in a mouse muscle, the fibers are much less stiff than the whole bundles. And this has important consequences for two reasons for me. One is I was very interested in the passive mechanical properties of humans. We were working mostly in Sweden with children who had wrist flexion contractures, and these muscles are extremely stiff. And we were trying to understand the source of the stiffness. And I'll talk about the sarcomere source in my next talk, but at least at this point, I'll show you the important first finding was that if you just here's a transverse section of human muscle. This is a typically developing child having sports surgery. This is a child, a wrist flexor in a child who has cerebral palsy, cross sections, and then longitudinal sections. You can see that the CP muscle just has a lot more ECM there. And again, I'm not an ECM person. I was in an orthopedic surgery department, so there were a lot of connective tissue biologists and biochemists, so that was good. And it was kind of initially exciting because when we would measure the modulus of the muscles, this is now semitendinosus and gracilis. I'll tell you, in terms of contracture in humans, we've measured soleus, gracilis, semitendinosus, flexor carpi ulnaris, and all these muscles share the same phenotype that I'm going to tell you about to different extents. The point here is that if you carefully measure bundle modulus in kids with CP versus age-matched controls undergoing ACL surgery, the kids with CP have a higher bundle modulus. And then when you measure collagen content by the hydroxyproline assay, they also have a higher collagen content. So we thought, okay, this problem with contracture is really a problem with collagen synthesis. But the nice thing is, as you all know, when you do high resolution muscle mechanics, you can make or try to make a correlation between the stiffness or the modulus of the specimen and its content. And I will tell you that this kind of graph is very typical for us. There's not a great correlation between the amount of connective tissue as measured by a collagen assay, even if you look at subtypes. And even if you look at crosslinks, right? Those are the most obvious things. They just don't correlate very well. And at this point, we started to say, you know what, maybe it's not the amount of the material, maybe it's the type of material. And so my colleagues and I sort of started, I would say this is now maybe 10-ish years ago, we started down the road of trying to understand the structure and function of the muscle extracellular matrix. This is work that Alison Gillies did as a part of her PhD thesis. And again, in the interest of time, I'll boil down the story to two things. One is, first of all, there wasn't much, I thought what I was going to do is go read about skeletal muscle extracellular matrix. There wasn't much in the literature. So we developed three ways to look at muscle. The first way was ordinary scanning electron microscopy. And of course, as you know, it's a very noisy system. But when we looked at the connective tissue that surrounded fibers, so we got these images essentially by unrolling pieces of muscle, fixing it, and then doing SEM. The endomysial connective tissue around fibers was really like a mesh. People had described this before. Peter Perslow had some really neat images of this, and it looks like a mesh, sort of like you would see in any structure where it's fairly isotropic, meaning that the expansion of a muscle fiber longitudinally or radially would be resisted. And we thought, okay, well, this is a good way of load bearing, but it's not very directional. But I'll tell you that I was very surprised when I saw in these images a lot of these longitudinal cables. We call them paramecial cables, and just like anything in science, people had already known about this for a long time, especially in cardiac muscle. But it looked to us like it wasn't so much that the amount of the connective tissue was varying between the muscles. It looked like these cables were varying. But the problem with SEM is, first of all, you just get a single snapshot. So doing quantitative measures is tough. Second of all, the tissue shrinks. And third of all, the tissue distorts in order for you to make these measurements. So we tried a couple of other methods to measure these cables. One was to use confocal microscopy, again, worked by Allison Gillies. So here you see on the left-hand image, here's one of these cables. And you can see they have some intrinsic structure to them. Because this is under a confocal microscope and it's using a type 1 collagen antibody, we could measure sarcomer length by phase contrast and then measure the cables just microscopically and just look at the relationship between the cable angle and the sarcomer length. And as you would expect, when the cable angle is high, sarcomer length is low. And so you get this picture of basically a cable rotating as the muscle is being stretched. It does elongate a little bit, but these cables have a lot of intrinsic stiffness, and that's part of another story. But again, so the good news about this method is you can easily attach a force transducer to this fiber and measure force. You can easily measure this image, but in order to get a fiber or even a group of a couple of fibers, you really tear up the muscle and you distort the mechanical connections. You can see this even as you know, when you dissect single fibers, you can literally reach in there with forceps and grab the cable and pull it off. So we then went to a third method, which is all these are very time consuming, and it hasn't been extremely productive work over the years. But this is work that was developed by Mark Ellisman. Mark is a brain structure person. He studies cerebellar function. And essentially what we did is modified a method of scanning electron microscopy, which is called block face scanning electron microscopy. So here you can see an image. This looks like a transmission electron micrograph, but it's not. It's a scanning electron micrograph. So here's a muscle fiber, muscle fiber, muscle fiber. Here is a piece of a fibroblast in section. Here are some capillaries, and then you can see a lot of connective tissue here in the interstitium. And on the right here is a three-dimensional reconstruction of serial sections. So what you can see is as you go through these images, these cables undulate. You see a lot of other structures in the extracellular matrix. But the nice thing about this tissue is this is perfusion fixed tissue. And so the perfusion fixed tissue is in its natural configuration. And you can trace all kinds of things in this muscle. And I would say to this point, the major findings of these kinds of studies are that these paramecial cables are very long. We've followed them for many millimeters. There's tremendous amount of undulation, and while you don't appreciate it in most electron micrographs of muscle, fibroblasts in muscle are pretty big, and they're often associated, like you can see up here, they're often associated with a paramecial cable. So the question is, in terms of numbers, how many are there and how are they organized? And so for that, we resorted again to yet another method, which is essentially good old-fashioned stereology, where you put a grid over high magnification micrographs. And so here you can see, now you can, because it's higher magnification, you can see one piece of a paramecial cable. You can see here's one collagen fibril by the white arrowhead. Here are some other fibrils. These are more cables. Here's the basal lamina, and you can see the peripheral nucleus of a fiber. And we can quantify using stereology the number of collagen fibrils that are, separate single fibrils, and the number that are in bundles. And when you quantify that, you see this. So this is now, again, I'm flipping back and forth between models, but this is children with cerebral palsies versus typically developing children. And you can see one of the major findings is that essentially the area fraction of fibrils that end up in a bundle is significantly increased. It's interesting that we didn't see a change in the single collagen fibrils, and we did see also an increase in the area fraction of fibroblasts. So it does look like there is fibroblast proliferation. It does look like these fibrils organize, but I'll tell you from other models that are a little bit less noisy, it looks to me like in the extracellular matrix there is some kind of equilibrium almost, because collagen turnover isn't very fast. There's some kind of equilibrium or maybe assembly process that's related to these collagen fibrils. So the last study I want to talk to you about is the next obvious question: who's making the collagen and why? And for that, we borrowed a model from a colleague of mine who studies liver fibrosis, where we essentially measured the cells in the periphery that are making type 1 collagen. And of course, on the right-hand side, this is a transgenic mouse. It's the Nesprin-Desmond double knockout, which many of you know. These are Nesprin and Desmond are cytoskeletal proteins, but for the purposes of this talk, I'm only using them as a way to create fibrosis. And so you can see all this green in the extracellular space of a knockout compared to the wild type, which just has one. The nice thing about doing these experiments is we can make these mice and then we can isolate and sort the cells that are making collagen. And so what you see here is that when you look at higher resolution, some of the cells are extracellular, as shown here. You can see you're outside the basal lamina in the lower panel, but in the upper panel, this is actually a cell inside the basal lamina, which, as many of you know, probably is a satellite cell. So we sorted on satellite cells and fibroadipogenic progenitors, and the leftover we just called fibroblasts. As most of you know, there's not a great marker for fibroblasts. So in the double knockout, that's what the DKO is. We did see an increase in the area fraction of fibroblasts. We also saw an increase in the percentage of cells making collagen, but it's not like one cell dramatically changes its properties and therefore upregulates collagen. It looks to me like all of these cells are making collagen at a fairly decent level. If you measure the amount of collagen made by fibroblasts versus satellite cells versus FAPs, the fibroblasts make more by far. But I was very interested to see that satellite cells make a tremendous amount of collagen, especially type 1 collagen. And if you take these cells and then look at gene expression by the cells, you see various signatures. So now here we are, here we're calling them skeletal muscle progenitors, so satellite cells, fibroadipogenic progenitors, and then fibroblasts. And you can see the various collagen isoforms. I've tried to sort of code them, but these heat maps are a little odd. The good news is that the cell types sort very well. In other words, they're very distinct expression profiles. It does look like, the expression profile of the laminar collagens ends up being more closely associated with the fibroblasts and FAPs compared to the skeletal muscle progenitors. We're just in the very early phases of this stuff, and I really don't know enough about collagen to know all the different organizations. That's part of the reason I would present a slide like this to this group, because I know you all know more about it than I do. But at least my take-home lesson for this is that these populations are regulated. I have some other data showing that, for example, if you delete one of the populations, the other two populations go crazy. So my current thinking is there's sort of an equilibrium in the extracellular matrix among the mononuclear cells that are responsible for making collagen. We don't really understand the specifics regarding who regulates them or how they do. The structure of the extracellular matrix is still yet to be determined, but I think these paramecial cables are going to turn out to be a big deal. We're trying to do studies to show that they are the appropriate load-bearing structures. And the jury is out on a lot of the other structural components, especially fibroblasts, because one of the sad tricks God has played on us is that fibroblasts are kind of hard to track.
4 minutes left.
Yep, okay, so I'll summarize here. Fibrosis is poorly defined and understood from a structure function point of view. Almost all of our biochemical assays do not correlate very well with our mechanical assays. And that's where we're onto this whole idea of other structures such as paramecial cables. Fibroblasts, satellite cells, and FAPs all collude, that's my key word, in the production of collagen-1 during fibrosis. We don't really understand the control or specificity. I think there's a lot to be learned. I'm still learning about FAPs in other models. And again, I know you all know more about that stuff than I do. And I think essentially new tools are required to understand the fibrosis in these models, either mouse or humans. One of the problems is in order to understand it structurally, you need nano level tools, but in order to correlate them with mechanics, you need to be able to study these things over many millimeters. So and the only method so far we've been able to determine that will allow us sort of nano levels, structural assays, and sort of micro level distances is that serial block face EM. And because it's an EM method, you get one single snapshot. So I'll stop there. I tried to leave a couple minutes for questions, but thank you very much for letting me present this.
Well, thank you very much indeed, Professor Lieber. This has been a really inspiring presentation as usual. And it's really a state of the art on the ultrastructure of muscle and a different way to look at it. So are there any questions from the audience?
Yes, I do have one.
Yes, please. Dr. Tal, please.
Hi, Dr. Lieber. Thank you very
Hi.
much for your presentation. I would like to ask you about the difference between a muscle disease such as a muscle dystrophy that is also being characterized by fibrosis and the cerebral palsy that you have shown, that seems to me to be, that has to do with the tension in the muscle, being spastic chronically. Mm-hmm. So do you have a difference between, can you see difference between the muscles, the cells that are responsible for the fibrosis in these two situations? This is one question. And what does the immune system, like local cells such as macrophages and others, have to do with the appearance of fibrosis in the muscles?
Mm-hmm. So the second question is a little easier than the first one. The second question, at least in the transgenic models, there's a tremendous amount of inflammation, local and systemic inflammation, whereas in CP, there's not. So I think, when we say fibrosis as muscle people, it probably is a common phenotype, but I think you can come up with fibrosis probably 10 different ways. And for example, Duchenne muscular dystrophy, the fibrosis is clearly, a satellite cell depletion, muscle degeneration, regeneration, massive inflammation, et cetera. In CP, I will tell you, in my next talk, I'll even show you, in CP, even though there is some spasticity, most of these contractures, they're fixed. By the time we see them for surgery, they're fixed wrist flexion contractures. They're really not spastic, meaning, for example, if you inject Botox, you don't release the contracture. It really seems to result from an epigenetic modification of the satellite cells in the muscle. And I'll show some of that a little bit later. So I think the take-home lesson is there's probably many mechanisms to produce fibrosis, and it all kind of looks the same when all is said and done. We can't label the cells of humans, so we're in the process of trying to understand in humans the experiment I showed you in mouse.
Thank you.
Thank you. And one last question.
Well, there is Carlo. Carlo, you haven't asked questions so far, so.
I asked last question. My last question was about satellite cells. I mean, you mean really quiescent satellite cells? I always thought about satellite cell when they are quiescent as completely inactive, but in reality, they seem to be able to produce something, no? At least not very many, but anyway, in the list of different collagens and molecular extracellular matrix, there was a little group, say 10, 20 type of molecules expressed by the satellite cells in their quiescent state. This is the question mark, in their quiescent state.
This is true. So the satellite cells we isolated there were MyoD positive, so they were activated satellite cells, almost certainly. Right now, in the transgenic model, sort of like a dystrophy model, these muscles are not very happy. Part of the reason they're fibrotic is I think their satellite cells are chronically activated. We have a little bit of evidence, like the Duchenne model, that in the Desmond-Nesprin double knockout, the satellite cell population is turning over rapidly. The vast majority of them are activated. So I can't say that a truly quiescent satellite cell is making type 1 collagen, but in this transgenic model, 80+% of the satellite cells are making type 1 collagen. It's strange. I'm surprised as well.
Yes. Something new, at least.
Very, very, yeah.
Maybe a very, very short question. Feliciano, did you have a question?
Very briefly, can you tell me 2 words about this cable you have described? Because I've seen immunofluorescence images and EM, and in my life, I've done some EM and some immunofluorescence of muscle. So it looks to me that your fibers were pulled apart.
Yes, yes. So in the confocal I showed you, I'm not saying that cable is how it is in the normal muscle. In fact, I don't believe it is. I think our dissection pulled it loose from the fiber. But when you look at them in the block face EM, they are in the extracellular space. What we were trying to see is, where do they connect to the fiber? Are there focal adhesions or whatever? We saw nothing like that. We saw so far in many millimeters of space along the fiber, we see these cables essentially free floating so far. We're trying to develop methods where we can get longer.
But also in the EM, I think your fibers were pulled apart.
Even in perfusion fixed tissue?
They were pulled apart. I mean, it was clear from the section.
Well, they were apart. I don't know. I guess the question is, were they pulled apart or do they live a little bit of apart? Because as you know, as an expert, part
usually,
of it...
No, usually in histology, you can see they are close to each other. They're not pulled apart like that.
Okay.
I think that was pulled apart in the EM preparation.
Okay. Fair enough.
This is very, thank you both.
Sure, of course.
Stimulating talk, and I think we can carry on later on. But
Yes.
for the sake of time, thank you very much again, Richard, and see you later.
My pleasure.